This is the third video of the ionotropic receptors lecture number 2. In this video, we will talk about glutamate receptors, specifically those related to AMPA receptors and Kainate receptors. So, glutamate is the most common excitatory neurotransmitter. Regarding glutamate receptors, there are two main groups or families of receptors. Those that belong to the ligand-gated ion channels, including NMDA, AMPA, and Kainate receptors, and those related to gene-protein coupled receptors, also known as metabotropic receptors. For this lecture, we will discuss those related to ligand-gated ion channels, which are also considered the ones that mediate a fast excitatory synaptic transmission. So, ionotropic glutamate receptors are composed by different subunits. They form a channel composed of 4 different subunits. It was originally proposed that each GluR subunit could have 4 transmembrane domains, but now we know that there are indeed only 3 transmembrane domains that we can see in this diagram. And then there is another part of the protein that gets inserted into the membrane, but it doesn't cross all the way around, and it's called like intermembrane loop, and this is also playing an important role in the formation of the channel. This each of these subunit has an amino terminal domain that is composed by the N-terminal part of the protein. Then it's followed by a ligand binding domain where glutamate binds or other agonists bind, and then the transmembrane domain followed by the carboxy terminal domain. This is the general structural feature for ionotropic glutamate receptors. This is how you can write it in a sequence what the domains of the protein are, starting for the amino terminal domain. S1 is the segment 1 that is related to one of the loops that form the ligand binding domain. Then the transmembrane domains with the intermembrane part. Then segment number 2 is also part of the ligand binding domain that it's interacting with S1 to form the ligand binding domain, then the M4, and then the carboxy terminal domain. This is how different subunits for the glutamate receptors look like, with the ATD at the top, and then the ligand binding domain and the transmembrane domains with this intramembrane loop. These M2 domains are the responsible for forming the pore, as we've seen in other receptors, with the transmembrane domain 2 forming the pore. In this case, it has the same function. It is not a complete transmembrane domain, but the functions of this segment number 2 are similar to other receptors. And there is a uniqueness in the ligand binding domain of the different subunits of the glutamate receptors. Some subunits can bind glycine, and some subunits can bind glutamate. So depending on how the subunits assemble, they're going to have affinity for both glutamate and glycine. Based on this, the glutamate receptors can be subdivided into 2 categories. One are non-NMDA receptors in which we can find the AMPA subtypes, Kainate subtypes, and then other types of non-NMDA receptors. In those, the ligand binding domain is exclusively binding glutamate. In the case of NMDA subtypes, then there is a combination of subunits that bind glutamate or glycine. And then we will see this in the next video talking about NMDA receptors. So now let's focus on AMPA receptors and the diversity of AMPA receptors. Um, it's huge. They mediate fast synaptic transmission. They're composed of subunits formed between GLU A1 and A2, 3, and 4, and they come from different separate genes. GLUA subunits, as usual, and other glutamate receptors have their N-terminal part facing the extracellular compartment, while the C-terminal domain is in the intracellular space. As I mentioned earlier, the ligand binding domain is made up from the N-terminal region, S1 and S2. And these are two different segments that come together to form the ligand binding domain. Its subunit, as I mentioned earlier, contains three transmembrane domains and one intramembrane loop. And then there are different specific domains, such as the flip-flop region and these other carboxy terminal region that are important for the diversity of the AMPA receptors. The diversity of AMPA receptors is increased by alternative splicing. All AMPA receptor subunits are susceptible to different forms of splicing variants. The carboxy tails of some AMPA receptors are also alternatively spliced in this part of the protein. There are also two modules, each containing 38 amino acids that can be alternatively expressed at a site immediately preceding the transmembrane domain number 4, called the flip-flop. In all AMPA receptor subunits exist two splice variants, their flip or their flop. The alternative splice cassette, it's also found in this C-terminal loop, and these places are susceptible to small changes. Only very few amino acids can change, but the effect can be quite dramatic, resulting in altered desensitization and different kinetics. So native AMPA receptor channels are impermeable to calcium, and this is a function that is controlled by the GLUA2 subunit. And just as an example of how these changes affect different subunits, the calcium permeability of the GLUA subunit is determined by the post-transcriptional editing of the GLUA mRNA, which changes a glutamine here represented by Q into an arginine represented by R. And this is the so-called QR editing site. So GLU-A2 subunits that contain the Q amino acid is calcium permeable, while the GLU-A subunits that contain the arginine amino acid are not. And almost all the GLU-A2 protein expressed in the CNS is the GLU-A2R form, giving rise to calcium impermeable AMPA receptors. These, along with other interactions with other intracellular proteins that interact through the carboxy terminal domain, provides a lot of versatility to the AMPA receptors. There are other types of splices, such as the R and G sites, that also confers different specificities in different types of forms. And in this slide, you can see a summary of how different types of editing affects different types of GLUR subunits, specifically for the AMPA receptor assembly. Here, as I mentioned earlier, the QR editing affects mostly GLUR2 or GLUA2 subunits and determines calcium permeability. Then the R and G editing affects mostly GLURs 2, 3, and 4 to variable degrees of editing, and edited receptors exhibit faster recovery from desensitization and desensitized to a lesser degree. Then the flip-flop splicing affects subunits GLU24, and flop isoforms is sensitized faster, while GLUR4 C-terminal splicing it's susceptible to phosphorylation and other modifications. So the AMPA receptors are responsible for the bulk of fast excitatory synaptic transmission, and there are like different mechanisms by which AMPA receptors activity induced increased conductance of the synapse. It's been suggested that they are either increasing the number of AMPA receptors at the postsynaptic surface by increasing inclusion, or modifying the existing AMPA receptors that are already there with different mechanisms. Um, Recent studies have suggested that indeed the overall conductance of a synapse is increased by receptor insertion rather than modification of existing receptors. And there are different proteins, specifically scaffolding proteins, that allow that to happen, that anchors the ionotropic receptors, the AMPA receptors, and also NMDA receptors to a specific places at the postsynaptic membrane. These receptors are characterized for a rapid reduction in the ligand-generated signal, which means that they're subject to a rapid desensitization after ligand binding. This is due to a conformational change that is induced by the ligand that also leads to its own inactivation. So this opening of the channel and sensitization is a very rapid process in the AMPA receptors. Then another one of the non-NMDA glutamate receptors is the kainate receptor, or also known as CARS. And these receptors were first identified as a distinct receptor type through their selective activation by the agonist kainate, which is a drug isolated from an algae. And I have to say that they were less understood than AMPA and NMDA receptors, and definitely less studied. Um, but what we know about kainate receptors is that they're ubiquitous in the central nervous system. This class of glutamate receptors is present at both sides of the synapse. Pre and postsynaptic kainate receptors can both regulate transmission at many synapses in a specific manner, and they seem to be involved in short and long-term plastic phenomena. So that highlights their significance for synaptic signaling. A curiosity about these kainate receptors is that those receptors located at the presynaptic surface have been implicated in inhibitory neurotransmission by modulating release of the inhibitory neurotransmitter GABA through a presynaptic mechanism. On the other side, postsynaptic kainate receptors are involved in excitatory neurotransmission. So, CARs in the brain form a tripartite hetero-oligomeric complex consisting of the 5 different GLUC subunits, GLUC from 1 to GLUC 5, and they assemble in tetramers and combine differently to form different tetrameric receptors that are also in close assembly, and they cooperate with this auxiliary receptor protein called NITO. Within the GLUC subunits, there are different kinds of affinity. There are low affinity subunits such as GLUC 1, 2, and 3, and high affinity subunits such as GLUC 4 and 5. And kainate type glutamate receptors restrict synaptic localization is determined by the afferent presynaptic connection. The mechanism that governs this impaired specific synaptic localization remains unclear. Recent studies have demonstrated that subunit composition in specific subunit domains contribute to synaptic localization of the kainate receptors. So, for instance, the cytoplasmic domain of the GLUK2 low affinity subunit stabilizes kainate receptors at synapses. In contrast, the extracellular domain of the GLUK4-5 high affinity subunits synergistically controls the synaptic specificity of kainate receptors through interactions with the complement protein C1q, as depicted in this diagram. So what this really means is that this input-specific synaptic localization of the native kainate receptors complex involves two mechanisms that underlie both the specificity of the different subunits plus stabilization of the receptor at different synapses. And then two other additional unique observations that make kainate receptors one of the most intriguing receptor family in the nervous system is that several kainate receptor knockout animals have been generated, but they have not been particularly informative about the function of these receptors due to the some lack of phenotype. And then some actions of kainate seem also to involve an unusual interaction with ionotropic receptors with G proteins. So this is a little bit more complicated and still like not very well characterized. And then the last thing about kainate receptors is that they're pharmacologically very difficult to distinguish from other glutamate receptors, such as the AMPA. But there are different molecules that have advanced in the last years to try to modify CAR receptors in a more specific way. And as usual, I leave you here with a short quiz where you can revise the most important concepts that we've seen in this video, and we will discuss these and other questions in class.